Regenerative capacity, as we all know, decreases with age. When we're embryos, it's superhuman. We can repair just about everything. And when we're young adults, it's still quite strong. We regenerate very quickly from muscle damage, bone damage, and we don't get these diseases of aging. But when we're older, we do, both on the outside as well as, more importantly, on the inside. So this regenerative capacity is in our genes. The question is, how do we turn it on? And how, by turning it on, can we potentially extend healthy human lifespan? That's Alevian's goal and mission. So the story of Alevian goes back about 15 years ago when my co-founder, Professor Amy Wagers, was a postdoc at Stanford. She worked on a Frankensteinian experiment called parabiosis, where they sew together a young mouse and an old mouse. And when they do this, the circulatory systems fuse between those mice. And you get these incredible effects. The young mouse experiences regeneration across just about every tissue and organ system that's been studied. And interestingly, the young mouse prematurely ages. So these experiments were initially reported in Nature magazine around 2005, and since have been repeated by lab after lab after lab, and you've probably seen a lot of this in the popular media, the regenerative effects of young blood. And some people think of a business opportunity that we're going to open up transfusion clinics and young people are going to give blood to old people. And there might be some opportunity here, but it's not a scalable therapeutic for sure. And our co-founders thought about a different approach. So Professor Wagers got together with another one of our co-founders, Professor Richard Lee, when she got a permanent appointment at Harvard University. And they wanted to figure out what factors in the blood are responsible for this effect. So they looked at all of the metabolites in blood, or everything that they could study. They couldn't find anything there. They looked at all of the lipids in the blood, and what they were looking for is there a statistically significant difference between the factors in the young animals and the old animals? They didn't find it in the lipids. And then they used a proteomics platform, a diagnostic on a chip that looked at 1,300 different proteins, and they found something. They found a little-known protein called GDF11 that was significantly different, about 2.5 times different between the young mice and the old mice. Now, what's fascinating about GDF11, we didn't know a whole lot about it at this point. We knew that in development it was absolutely critical. If you knock it out in mice, they're born dead with failed kidneys, with malformed spleens, pancreases, vertebra. We also knew that this protein is the exact same in its mature region between mice and humans, excitingly. So they took that protein and they injected it in aged mice. And much to everybody's surprise, they were able to show the same regenerative effects that you get when they're sewn together. So the first report that came out was in Cell at Professor Lee's lab, and it showed that only 4 weeks of GDF11 remodeled the heart to a younger state. So it actually repairs, doesn't slow aging, repairs, regenerates the heart, and also the biomarkers of heart disease. Number two, in Professor Wagers' lab, she was able to show that it stimulates the same rate of repair to skeletal muscle as we have in our youth. Gosh, I could use this. Number three, in another Alevian co-founder's lab, Professor Lee Rubin, a neuroscientist, he was able to show that GDF11 caused new blood vessels to grow in the brain and new neurons to form in key areas of the brain. So when these came out, you can imagine this big, phenomenal surprise. One of the breakthroughs of the year in Science magazine. Now, since this point in time, this represents our original studies. There have been a whole series of studies that have come out showing lots of regenerative effects. There have been some other effects that I'm happy to talk about with any of you, also that are a little bit contradictory, that took a little while to clear up. and we think we really understand them now. So talk about what we're trying to do. So GDF11, we know at least its active form appears to go down in humans and animals with age. So one approach is just to supplement GDF11 itself. To the best of our knowledge, it requires daily injections. We use daily injections, and we see effects in as little as 9 days. We're also working on variants of GDF11 that we hope to produce that have even better properties, can dose less frequently. Now, we've also discovered a degenerative factor that goes up with age. It binds to GDF11 and takes it out from functioning. So another strategy is to knock down with antibodies that degenerative factor. Perhaps the most interesting thing, because this is the same protein in mice as it is in humans, there have been some data. We haven't yet tested it in humans, but we've looked at levels of this protein in humans. Actually, a lab out of UCSF did. What they found is the people that have the lowest levels, the lowest 25% level of GDF11 compared to the highest 25% level, have much higher risk of dying over a 5-year study period, and much higher risks of heart attacks, heart failure hospitalizations, and strokes. So it is actually predictive. If you measure them at the beginning of the period, it's about as predictive as if you told me you smoke 2 packs of cigarettes a day to have low levels of this protein. That would suggest, we would hope, that when we give it to humans, it will have an incredible regenerative effect. Now, we also found very similar effects with the antagonist, the degenerative factor. It is also predictive. High levels of this factor are predictive of death and disease in humans, with the goal to ultimately take us to a much healthier, longer living place. With that, here's my contact information if you have any questions for me later. Thank you very much.